Hello everyone, and welcome to my presentation. My name is Dr. Csengele Barta. I am an associate professor of biology specializing in plant molecular ecophysiology in the Department of Biology at Missouri Western State University. And my background and research interests involve those mechanisms that plants use to interact with their environment. So today's presentation will focus on a very specific interaction, specifically the chemical warfare plants are waging on their unsuspecting neighbors, in particular how plants use allelopathic metabolites and secondary metabolites in order to compete with their neighbors, mechanisms that are very important, for example, in the case of invasive species that use allelopathic inhibitory chemicals, exudates, or volatile metabolites that are released into the environment that can inhibit the germination of seedlings of native species, or they can inhibit the growth as well as the development or even the survival of some of those native species, therefore making the invasive species particularly successful in environments into which they have been introduced. And this poses several problems in terms of loss of biodiversity, loss of native vegetation, as well as challenges conservation efforts and management efforts of many of those invasive species. So today's focus is going to fall on some of these interactions and our work that aims to uncover and study the mechanisms behind these interactions in order to potentially develop better control strategies, as well as to better understand in general plant-plant interactions. Just to give you a short overview of what we are going to talk about, I will address the complexity of plant-environment interactions in general, and then I will focus on the chemistry, the secondary metabolism that plants use specifically to compete with their neighbors, and we will address a very specific group of chemicals that are called allelopathic chemicals that have negative impacts on species in the native environment of a variety of different kinds of invasives. We will talk about one invasive species in particular, which is Amur honeysuckle, the Lonicera myki, very widely spread across the Midwestern United States, which releases phytotoxic chemicals that alter their microenvironment, being toxic at multiple trophic levels. And I am going to show you some of the studies and preliminary results on the inhibitory effects of honeysuckle leaf extracts on the germination of a model species seeds, Brassica, that we have used as a model to study these mechanisms in my lab. And I will close by discussing the future directions and open questions that we will be addressing in our future research in my laboratory. Now, when we think of plants, we also have to consider that plants are not independent of their environment. They are in very strong interaction with their environment, and that includes on the one hand the abiotic environment. They depend on factors of the abiotic environment because their life, for example, depends on the energy of the sunlight. It depends on the precipitation, the amount of precipitation that they receive from the environment, depend on the optimum temperatures. They are affected by changes in the temperature. They are exposed to the surrounding air. The pollution, for example, has a strong negative effect on plant physiology and biochemistry. Elevated carbon dioxide has an effect on plant growth and development, as well as they also interact with many of the factors that are below ground as well, such as soil chemistry, soil, water, nutrient availability in the soil, and many others. So plants depend on the plant's optimum physiology, depends on the environment, depends on the environmental abiotic non-living factors, but in the same time plants also feed back on that environment, and many plants release chemicals into the environment that can be volatile chemicals or can be non-volatile exudates that feed back, for example, on some of the abiotic processes, on air quality, on overall changes in the environment, such as, for example, the resilience of greenhouse gases in the environment, therefore indirectly also feeding back on processes that relate to a warming or a changing climate. Therefore, this interaction is a bidirectional interaction with plants depending on their environment, but also providing feedback onto the abiotic environment. And the same types of ideas are also valid in case of a plant's interaction with its biotic environment. So the living factors in that environment, which can include, for example, its interaction with herbivores, its interaction with pollinators, interaction with parasites of the given herbivore, with which plants can control at multiple trophic level, for example, biological invasions, interactions with other biotic factors in the environment that potentially can enhance protection of the given plant, including its defense mechanisms, interaction with the soil microbiome, interaction with fungal agents, and very importantly, interactions between plants as well. So plant-plant interactions are also part of this biotic environment interaction that plants control through a very elaborate chemistry, secondary metabolism, that allows them to perceive signals from both the abiotic as well as the biotic environment, process those signals, react to changes in the environment, as well as feedback onto both the abiotic and biotic environment. In the same time, in addition to those interactions between the plant and its environment, the abiotic environment also has an impact on the biotic environment, as well as the biotic environment can feed back on the abiotic environment as well. Therefore, these interactions are extremely complex and can occur both above as well as below ground. When we consider the plant organism influences the development of plants, in which phytohormones play a critical role, as well as affects their primary metabolism, for example, metabolic reactions as photosynthesis or respiration, as well as secondary metabolism, which includes a variety of molecules belonging to a variety of metabolic pathways that generate secondary metabolites belonging to the large group of terpenoids or phenolics or alkaloids, affects the oxidant-antioxidant balance in plants, and the totality of these secondary metabolic pathways strongly contribute to defense, adaptation, and the adaptation reactions of plants, as well as they play a critical role in the communication of plants with their abiotic as well as their biotic environment. And today we will specifically focus on a portion of this very complex picture that involves the interaction of plants between each other. So plant-plant interactions specifically in in competition with each other, so the competitive interactions that will involve, again, a group of secondary metabolites with which they can actually control the survival, the germination, growth, development, and survival of their native interacting partners. Now, when we consider these interactions, and when we focus specifically on the plant-plant interactions, especially important in competition for resources, we know that plants, for example, or either living organs, above ground organs of plants, or the degrading or decomposing organs of plants within the soil, such as decomposing leaves, can emit a variety of biogenic volatile organic compounds that can have an effect on neighboring plants of different species, that can, for example, delay or inhibit the germination, the growth, and alter the development of those other plants in the environment of the plant that's considered to be the donor plant, in terms of whenever these plants are competing for a particular resource in their environment. In the same time, even below ground, these interactions become important and critical, because plants can release a variety of exudates and leachates that leak into the soil, and they can also release volatiles below ground. All of these can play a role even in the below ground region in controlling, for example, the physiology and metabolism, growth, and development of a specific target species in the environment of the donor species. So there is a lot of chemical detail behind these interactions, and plants are able to synthesize hundreds and hundreds and thousands of chemicals that can be either volatile or non-volatile, and those chemicals can significantly affect, they can affect negatively, some of them which can affect even positively the neighboring species, which affect their competition for a limited set of resources in their given native environment. And those effects can occur at a variety of different levels in terms of the life cycle and the physiology and the metabolism of the target plant. It can be observed at the level of germination or growth, or alterations in the development, changes in the reproduction of the given species, but can all impact the survival of the target species. Now, there are lots of factors that can influence the success of all of these interactions. And on one hand, for example, when we look at whether those chemicals that are released either above or below ground the plant can actually realistically impact the target plant, we have to consider the distance that these chemicals have to surpass in order to reach from the donor plant to the target plant. The target plant responses might depend on the amount of these chemicals in the environment, so specifically the concentration. It might depend on the properties, specifically chemical properties of the released secondary metabolites. That includes, for example, volatility in the case of the volatiles, as well as the solubility of non-volatile substances that have to be solubilized in the soil water after degradation of the given organ in the soil. There are lots of factors that are soil dependent factors that can influence the success in these interactions. For example, the organic matter in the soil, the texture or the structure of it, the moisture content, so the water retaining capacity of the soil can alter the chemistry and the chemical success as well. whether there are organisms in the soil that can for example break down those chemicals that are released from the donor species. The pH, the acidity can significantly alter again the chemistry behind these interactions, or the ion exchange capacity of the soil particles. So there are lots of factors that can affect, for example, below ground interactions significantly. Now, some of these chemicals might be retained on soil particles. They can be transformed, so microbial transformation of donor plant released chemicals is pretty common, or they can be transported. They can be transported successfully with soil water to a target plant, in between the air particles in the soil, for example, below ground, and through diffusion as well. And then there are lots of factors that are actually plant-dependent factors that will define ultimately the response of the target plant, and that depends on the species, on the variety, depends on whether the exposure happens in a particular growth stage. So it depends on the phenology, it depends on the affected organ, and also depends on physiology. It depends on the health of the overall health of the plant, and a variety of other factors as well. And ultimately, most of these interactions will also be controlled by environmental factors. They will depend on temperature. These processes are temperature dependent, precipitation availability dependent, air chemistry dependent in case of the volatile signals in the air, can be dependent even on irradiation, or for example, in the case again of those volatiles above ground, and the photooxidation of some of those compounds. So there are a lot of factors that ultimately influence how successful an allelopathic interaction is going to be in between a donor plant and a receiving or target plant, and how those impacts will translate into changes in the germination, growth, development, reproduction, and ultimately the survival of the species that's being exposed to this really large array of chemical attacks. Now, one interesting question that we all have right now is the environment and the effect of the environment, not just on the chemistry and then the transmission of these molecules, but also on the overall physiology and the interactions of both a donor type of plant as well as a target type of plant with the environment itself, in a changing climate, in a changing environment that's expected to bring a lot of changes in these environmental factors, including sunlight availability, or the spectrum of the received irradiation, changes in the temperature, changes in air chemistry, particularly an increase in the amount of carbon dioxide in the air, or an increase in the pollution levels, decreasing amounts of precipitation, uncertainties that lay on the severity of these changes that can be expected both in short term as well as long term in the environment, that will ultimately affect the plants themselves as well as their interaction with their environment. So as an extension, it will also affect the interaction between the allelopathic interactions or interactions between individual plants, and will affect down the road the competitive processes in between plants as well. So there are lots of open questions today that target these responses. Therefore, better understanding of the basic mechanisms that lie behind some of these processes are critical in order to be able to understand how this will translate down the road to a future environment that will present additional challenges in terms of many changes and concurrent changes in the environmental factor effects as well. Now, allelopathy in plant-plant competition for resources has been shown to be a very effective tool in the arsenal of many plants, specifically invasive species that are introduced into areas where they do not have specific natural enemies, and using allelopathy and using chemistry, using volatiles or using non-volatile chemicals that are released either to the soil or into the air is a very good mechanism that many invasive species take advantage of. And just to show you a couple of specific examples related to this, for example, the spotted knapweed that actually has an Eastern European origin, and it has been imported into the United States from Romania, my home country, releases into the soil catechin, and the catechin enrichment in the soil has a drastically negative inhibitory effect on the native vegetation in the area where the spotted knapweed takes resilience, therefore restricts significantly the growth and germination of many native species. Now, these allelopathic chemicals will have, as you will see, I selected a couple of examples that show specifically that these chemicals can be of different nature, catechin being just one of them, but most of you might be familiar with the effects of the decomposing black walnut leaves and walnut husk in the soil that releases this chemical that's called juglone, which is highly toxic, again, on the germination and growth of many species nearby the black walnut. Juglone, for example, is a very important toxin that most recently in medical science is being evaluated as a chemotherapeutic agent because it inhibits for example, not just the cell divisions in a neighboring plant, but it can have similar effects even on the animal organism as well as the human organism. While on the other hand, the catechin also inhibits cellular divisions in the meristematic regions of native plants in the area, but it also triggers a variety of reactive oxygen species and oversynthesis of those reactive oxygen species, which physically damage the cells as well. Therefore, they lead to cell death in meristematic cells. The broomcorn or sorghum releases sorgoleone, for example, and these species, for example, have been shown to not just again inhibit other species in their immediate environment, but they can also affect biological denitrification processes in the soil. So they have an extended effect beyond just the plants that are in their environment, but they can also affect multiple trophic level and multiple biotic and abiotic factors in their environment as well. Now, another example to the release of allelopathic metabolites is the red fescue, for example, that actually releases a molecule that's a non-protein type of non-protein amino acid that has a very serious inhibitory effect on the cell wall assembly. Therefore, it affects nearby species growth by decreasing the potential for for cell wall synthesis and assembly, causing again very significant alterations in the growth and development of species, other species, native species in their environment. So overall, at many levels, these allelopathic interactions involve effects that are even beyond the simple interactions between plant and plant competing for resources, but rather they extend to interactions at multiple trophic level, affecting the soil quality, affecting the soil microbiome, affecting plants, affecting therefore through affecting the native vegetation. They can also affect the subsequent trophic levels. They can affect the herbivores of the native vegetation, pollinators of the native vegetation. So they have some really significant ecological impacts on overall specifically on these smaller microenvironments that surround these particular introduced species. Now, one of the species of interest specifically in Missouri and in the Midwest is Amur honeysuckle, or Lonicera mackey, because Lonicera mackey is one of those introduced invasive species that very quickly took over the Midwestern areas, and it's actually spread beyond the Midwest to other regions of the United States as well. Amur honeysuckle is native to Eastern Asia, and it has been introduced into the United States in the end of the 1800s, specifically in 1896, as many other invasives have been introduced into the US as well. For reasons of being used as an ornamental species, it has a very, very good, interesting ornamental quality. And it has also been partially introduced with the goal to minimize soil erosion at the beginning of the century, as well as being used as wildlife cover as well. Nevertheless, when we look at the anatomy and the morphology of Amur honeysuckle is shrubby. It's a very dense shrub with erect branches that can be up to 30 feet tall, so it can grow even higher than a human individual, and essentially it can populate the understory of a forest environment really, really quickly, especially in undisturbed environments, because it provides such a dense cover, essentially shades and eliminates the potential for native understory vegetation to grow and thrive in this environment. Its seeds are spread by a variety of species that include birds, mammals, therefore it can very quickly spread. Interestingly, this species has a really, really high resistance towards a variety of environmental conditions. It can resist really well cold temperatures, sometimes doesn't even die back at the end of the season, and it can resist also high temperatures. It can thrive on any kind of soil. It does well with low as well as high precipitation. So essentially, it's really, really hard to control the species and poses specific challenges in its control, partly because of its resilience, as well as very, very quick spread mechanism that is used that the species can spread with using biotic agents to disperse their seeds, as I said earlier, using mammals or a variety of bird species. Now, forming this extremely dense and hard to penetrate understory, it has multi-level ecological impacts, not just on the vegetation, the native vegetation, but also poses a mechanical, physical obstacle, for example, for the migration or movement of animals as well. in the biotic environment, because it forms this really dense vegetation cover. And what has been recently observed in a couple of studies by Don Cipollini and his colleagues is that Amur honeysuckle leaves degrading in the soil at the end of the growing season actually lead to the release of a variety of phenolic metabolites within a metabolic profile being dominated by apigenin and luteolin, and their glucoside derivatives. But these chemicals have a devastating chemical effect as well, a phytotoxic effect on herbivores, on the soil microbiome, as well as have the potential to inhibit the native vegetation as well, in addition to all of those other physical disturbances and ecological disturbances that I mentioned earlier. Now, this obviously drew a lot of interest from my laboratory, because my lab and my research interests are particularly invested in understanding the mechanisms that invasive species are using to compete with the native vegetation, and therefore we initiated a project not so long time ago in which we study the inhibitory effect of the same honeysuckle leaf extracts that Cipollini has described in a larger ecological context earlier on the actual mechanism of germination in model brassica seeds. And some of the research questions that we addressed initially were whether the extracts of the Amur honeysuckle leaves will inhibit the germination and or growth of the other species seeds and seedlings, in the first stage focusing on models as brassica, and whether the observed inhibition of this leaf extract is concentration dependent, so whether the impact of the synthesized chemicals is actually eliciting a different response at different concentrations. And what we have found is that indeed the Amur honeysuckle leaf extract did inhibit in a Petri dish plate assay the germination of brassica seeds, as you can see in some of these images, and the inhibition occurred in a concentration dependent manner, with the higher concentrations, for example, the concentrations as high as 2 grams leaf per milliliter in the extract initiated a completely lethal effect when seeds did not germinate at all. Nevertheless, lower concentrations only delayed the germination. So what I'm showing you here is a 24-hour germination rate that was about 100% in the control untreated seeds. Nevertheless, was progressively less and less in seeds exposed to different concentrations of the honeysuckle extract. Now, the next question that we asked ourselves was, are there actually any seasonal variations in the accumulation of these inhibitors in the leaves? And what we have found by testing, extracting, preparing aqueous extract of Amur honeysuckle leaves throughout a full growth season starting in March and ending towards November, using a non-lethal concentration that we established earlier at 1 gram per milliliter in the same 24 hours germination plate assays. What we found is that the extract becomes more and more inhibitory, delaying germination, especially towards the end of the growing season. Therefore, closer to the abscission of those leaves from the shrub itself. Therefore, they tend to accumulate towards the end of the season, starting with August through November. Higher and higher concentrations, higher and higher amounts of those inhibitors in the leaves, leading to a complete inhibition in derived from the extracts that we prepared from leaves harvested in November. Now, another question, obviously, that we asked immediately was, what are the actual mechanisms or potential mechanistic routes behind the observed inhibition, and whether there are any ways this inhibition can be relieved or partially relieved or even delayed. And when we look at how seeds germinate, the germination process involves a very complex metabolic signaling pathway that essentially starts with the imbibition of the seed, water uptake. That could be potentially one of the mechanisms that could be impaired, could be a target mechanism that could be impaired by the chemicals in the amur honeysuckle extract, by inhibiting some of the aquaporins that transport water, or a second target mechanism that we were even more interested in is the second step within the initiation of germination, and that is the synthesis and release of gibberellic acid, which is a phytohormone within the embryo's body that activates an entire signaling cascade pathway that essentially activates the enzymes that break down the food resources that are stored in the endosperm, so specifically leading to the synthesis and the activation of amylase that breaks down the starch, releases the sugar reserves that are required for the subsequent growth of the embryo within the seedling, and leads to successful germination and eventually the growth and development of the seedling itself. So one of the mechanisms that we became particularly interested in, that we hypothesized that could potentially be significantly inhibited by the chemicals that are released by the amur honeysuckle, could be potentially the mechanism that impacts gibberellic acid synthesis and release in these seedlings. And in order to test that, we have employed brassica varieties which had altered gibberellic acid production. Our control was a standard seed that produces physiological amounts of gibberellic acid, that's also abbreviated as GA3, a variety that's called the tall variety that produces 10 times higher gibberellic acid than the standard control variety, and gibberellic acid deficient dwarf variety that produces 4 to 10 times less gibberellic acid than the standard variety. Now, when we compare the phenotype of these plants, we can see that they are very different. They all germinate. They are somewhat similar. The dwarf is a bit shorter than the standard, and the tallest is a little bit more slender when it reaches a 7 days age, and then they are significantly different in their mature and reproductive phenotype as well, because one of the functions of gibberellic acid in plants is to trigger or to aid the germination process, and also causes the elongation of the plant body, causing higher distances between the nodal regions, so it increases the internode length, and that's pretty obvious when we compare the mature 14 days old plant of the standard to that of the tall. Now, the one that's deficient in gibberellic acid shows a much much shorter phenotype. It's dwarf. It has much shorter internode length as the standard variety, and of course much shorter than the tall variety. And here you see 72 hours germination pictures of all three of these varieties. Now the fact that we have varieties that are gibberellic acid overproducers or gibberellic acid underproducers helped us in comparison with the standard to actually compare how the differences in the endogenous gibberellic acid concentration actually affect the response of the plant to exposure to the inhibitor in a plate assay. And what we have found is that compared to untreated controls, the standard germination in the presence of the honeysuckle extract, the non-lethal amount of honeysuckle extract, the germination was delayed significantly in the standard variety. Now, when we looked at the gibberellic acid overproducer plants, we found that even though their germination was also delayed, it was not as much delayed as it was in the standard variety. And then in the case of the dwarf or gibberellic acid underproducer plants, these plants treated with the Lonicera extract did not germinate within 72 hours at all. So it completely inhibited the germination of the dwarf plants that were gibberellic acid deficient. So we can say that metabolites in the decaying leaves of the invasive amur honeysuckle negatively impact and delay the seed germination of brassica, but the inhibitory effect of the honeysuckle extract can be relieved in those cases where more endogenous gibberellic acid phytohormone is available internally in the seedling, and that is the case of the tall plant, where the inhibitory effects were the least in this particular germination seed assay. Now, we were also interested whether gibberellic acid, specifically exogenous, externally added gibberellic acid, triggers and relieves these negative effects on germination. And we tested all three varieties, and we found that in the standard plants, the presence of additional externally added gibberellic acid actually speeds up the germination of the standard variety. Therefore, the inhibitory effect is a lot less accentuated than in the case when the plant was not treated with exogenous gibberellic acid, we observed a very similar situation, perhaps even less delay, even in the case of the tall plants that were originally endogenously also more enhanced in gibberellic acid concentration. And here is where we observed the biggest difference, and that's the case of the dwarf plants, which are gibberellic acid deficient. Remember, in these plants, the Lonicera extract alone inhibited completely the germination. Nevertheless, when the Lonicera extract was also accompanied by exogenous gibberellic acid enhancement, that was able to relieve to some extent, even though it was still delayed, but it was able to relieve by the 72 hours benchmark the complete inhibition of the seedling germination. Therefore, the exogenous gibberellic acid also had a stimulating effect on the germination, or it relieved the germination inhibitory impacts of the Lonicera maici extract. So the inhibitory effect of the honeysuckle extract can actually be relieved by exogenous gibberellic acid phytohormone enhancement. And when we compare these responses that we observed, again, the exogenous gibberellic acid clearly decreased that gap in between in the delay of germination in the standard seedlings with physiological concentrations of gibberellic acid. Its impact was even higher, and the exogenous gibberellic acid enhancement impact was the highest in addition to the endogenous in the tall plants. And then it actually, the exogenous gibberellic acid addition allowed the dwarf plants to overcome the inhibition caused by the honeysuckle leaf extracts. So overall, the effect that we observe is strongly dependent on the plant's ability to produce gibberellic acid endogenously, as well as it can be relieved to a partial extent by exogenous gibberellic acid treatment as well. Oops. We also tested the impact of these extracts on the growth or development of the radicals of the seeds that have been exposed to the Lonicera extract and had a delay in their germination. And what we found is, again, the most important effect that we observed was in the dwarf plants that were deficient in endogenous gibberellic acid production. But when the gibberellic acid was added to these plants externally, then the radical growth has been observed, and therefore the negative effect of the honeysuckle leaf extract was relieved, while the effects were much less pronounced in the case of the standard and the tall variety that was overproducing gibberellic acid. And as far as the hypocotyl length and extension is concerned, we did not observe significant impacts of neither the endo or the exogenous gibberellic acid in neither one of those varieties, at least not at this early stage of the plant's growth and development. So what we concluded was that metabolites in the decaying leaves of the invasive Amur honeysuckle negatively impact radical and hypocotyl elongation in the brassica seedlings, but the inhibitory effect of the honeysuckle extract can partially be relieved by gibberellic acid phytohormone enhancement, with dwarf plants benefiting the most on short term from the phytohormone enhancement. So overall, we can conclude in a preliminary manner that the aqueous Amur honeysuckle leaf extracts do inhibit and or delay brassica seed germination in a concentration-dependent manner, that the accumulation of the allelopathic inhibitors and the negative allelopathic potential of Amur honeysuckle leaf shows seasonal variation with increases in the end of the season specifically. Metabolites in the decaying leaves of the invasive Amur honeysuckle negatively impact and delay seed germination and seedling growth in the brassica, and that the inhibitory effects of the honeysuckle extract can be relieved by endo and or exogenous gibberellic acid phytohormone enhancement, benefiting in particular plants with low endogenous hormone production. Now, this project is far from being finished, and through the course of our investigations opened even more questions that will define the future directions into which my laboratory is going to go with this project. And one of the first things we would be interested in pursuing is to actually identify the molecular nature of metabolites that are responsible specifically for the inhibition of the germination And based on the previous findings of Don Cipollini and his colleagues, some of the candidate molecules that we will be testing in plate assays are going to be epigenin and or luteolin or their glycoside derivatives, because those molecules were the predominant components of the decaying aqueous extracts from the leaves. We would like to identify the primary molecular targets of the inhibition, because our assays to date have been only short-term investigations. Actually, we are interested in assessing the long-term effects of the inhibitor or inhibitors on seedling development. Assess whether there are differences in plant response based on the phenology of the plant at the time of the exposure. Our current experiments only targeted seeds and germinating seedlings, but that could include stages as seed, seedling, as well as mature plant in vegetative and or reproductive stages, and assess the potential uptake and transport mechanisms of these chemicals that we will identify as the major drivers of the inhibitory responses. Assess whether the inhibitors could be neutralized by selected candidate exudates released from the roots of native species in the area to allow for biological control, specifically the prevention of the inhibitors targeting other native species seeds. And this is particularly important because it would allow us to develop strategies that go beyond what is currently used by the conservation agencies to control the Amur honeysuckle, which includes chemical herbicides, controlled burning, as well as manual removal. Nevertheless, biological control and control using other species in the vicinity of the honeysuckle stands that would prevent the negative impacts of their phytochemicals reaching the target plants could be a very effective way to control their negative impact on the native vegetation. Also assess the impact of a warming climate on the Amur honeysuckle's allelopathic impacts on Missouri native plant species, and this is particularly important because, as I mentioned earlier, many of the factors in the abiotic environment are expected to change in the future to a yet unknown extent, and these changes will occur concurrently, and understanding the impact of the warming climate on the Amur honeysuckle itself, as well as its target partners, as well as the interactions between the two, would be extremely important in order for future modeling of vegetation distribution, and as well as predicting the future success of invasives as the Amur honeysuckle. And it would be very important for better understanding and developing mechanisms with which the impact of the honeysuckle could be minimized or decreased in the environment of the native species, as well as would open the door to developing better control strategies and control mechanisms that would allow for better actual control of the invasive species itself down the road. So that being said, I would like to thank to a lot of people that worked on this project, and that includes many of my students who worked in my lab. I would like to emphasize particularly Brian Jenkins. This is Brian. He was the ideator of this research, and his interest actually drove this research in my laboratory. And then the other students who worked on this project and continue working on this project: Lauren Tinoco, Kaitlyn Atkins, Harrison Meers. There is Harrison, this is Lauren. I don't have a picture of everyone, but some of them. Devon Lindstrom, Devon is here on both of these pictures. Rachael Prawitz, very long time research student in my lab who recently graduated. Zachary Shank, also graduated. Faye Tamarat, she also recently graduated. Cameron Hall, Amy Haddock, I don't have a picture of Amy, but she currently works in my lab. Jessica Pausch, Elika Zen, Caroline Langley, Ty McQueen, and Kaylee Van Guyson. They are all my current research students who work on this project. In addition, I would also like to thank to my dear colleague and friend, Dr. Kerry Chevalier, for the very, very extensive discussions on conservation issues and invasive species management that helped a lot in ideating and following up with this project. All of my colleagues in the Department of Biology at Missouri Western State University for the fruitful conversations and the monetary support specifically from the Program of Research, Teaching and Applied Learning, aka Portal, at Missouri Western State University, as well as research support from the Missouri Western Foundation for some of my students to present their research at meetings, at a variety of forums nationally and internationally that also is part of what forwards this project and gives us ideas and the opportunities for fruitful conversations on the future directions within this particular research line. So I would like to thank everyone, and I would like to thank the audience for listening to my presentation. And if you would have any questions, please do not hesitate to contact me. This is my information. I'm an associate professor in the Department of Biology at Missouri Western State University. This is my mailing address, and you have here my email. Please do not hesitate to address any questions, comments, critique that you would have related to this work. Thank you, everyone.